Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here. This is Andrew and Renee. Welcome to the Stack series. Today we are exploring reactive oxygen species, also known as ROS.
And it's a topic that really completely overturns a lot of what we think we know about health.
It absolutely does. We have a massive stack of cutting-edge biological research, molecular studies, and medical reviews in front of us today. And our mission is to uncover what is, quite frankly, a complete paradigm shift in how science views aging, disease, and cellular health.
Right, because for decades, you and I have been sold a very, very specific story about how our bodies age.
A story told on the back of literally every vitamin bottle, every expensive skincare cream, every green smoothie menu you see. We've been told that free radicals and oxidants are the absolute villains.
Sort of microscopic rust.
Exactly. The microscopic rust that causes our cells to decay, our skin to wrinkle, our bodies to break down. I mean, we spend billions of dollars globally to fight this microscopic enemy. We really do. But looking at the research today, we have to ask a question that turns that entire massive industry on its head.
What if these supposed villains are actually the very signals keeping you alive? What if, in our rush to extinguish the fire, we're actually cutting the communication lines of our own biology?
It is a profound paradox.
Right.
And to understand where the science is going, we really have to deconstruct this myth that all oxidation is inherently toxic.
Which is going to be tough because it's so ingrained in us.
It is, yeah.
Yeah.
But the literature we are looking at today, it spans from the fundamental chemistry of aerobic metabolism to the absolute frontiers of precision medicine. The story these papers tell is not a simple cartoon of good versus evil.
Right.
It is an incredibly nuanced look at how the very oxygen that allows you to breathe, think, and move is also operating as a highly tuned, deeply necessary, double-edged sword inside every single one of your cells. We really have to rethink everything we thought we knew about health.
Okay, let's unpack this. Because to really grasp why this is such a massive shift in scientific thinking, we need to start at the very beginning. We need to look at the foundations of what we think we know about oxidative stress. I want to build a baseline for you listening at home. Break down for me what these reactive oxygen species, these ROS, actually are on a molecular level. Where are they even coming from?
Let's travel inside one of your cells, specifically into the mitochondria. You often hear them referred to as the powerhouses of the cell.
Which is the classic middle school biology definition.
Exactly, which is fine for middle school, but a more accurate way to think about them is as highly complex, incredibly efficient microscopic engines. For these engines to generate the energy you need to live, a molecule called ATP, they rely on aerobic metabolism.
Meaning they need oxygen.
Right. You breathe in molecular oxygen, O2, and your mitochondria process it through an intricate system called the electron transport chain.
The electron transport chain. That sounds like a microscopic assembly line.
Think of it exactly like a bucket brigade. In this brigade, specialized proteins are handing off electrons down the line, one by one. The final person at the very end of that bucket brigade is the oxygen molecule you just breathed in.
Okay, so oxygen is the catcher at the end of the line.
Yes. Oxygen catches the electrons, binds with hydrogen, and safely becomes water. It is an elegant, life-sustaining process. But here is the catch. Just like any mechanical engine or any human bucket brigade, the system isn't 100% perfect.
There are accidents.
Right. It is highly efficient, but there are always minor leakages. Sometimes, an electron slips out of the hands of the proteins before it reaches the end of the line. It goes rogue, and it attaches itself to an oxygen molecule prematurely. When that happens, you've created a reactive oxygen species.
So it's essentially a byproduct, an exhaust fume from the engine of life?
That's how it was originally viewed, yes.
Yeah.
And there isn't just one type of ROS. The primary one that kicks us all off, the immediate result of that leaked electron, is called the superoxide anion. Chemically, it's an oxygen molecule with one extra unpaired electron.
And from what I remember of high school chemistry, electrons really do not like being alone. They want to be in pairs.
They absolutely demand to be in pairs. When an electron is unpaired, the molecule becomes a free radical. It becomes highly unstable and incredibly reactive. It will aggressively tear an electron away from any nearby molecule just to stabilize itself.
Just ripping parts off of whatever is next to it.
Precisely. If we want an analogy, think of the superoxide anion as a rogue, messy spark flying out of that cellular engine. It is chaotic, it is highly reactive, but because it is so incredibly desperate to react, it has a very short half-life. We are talking about a millionth of a second.
Wow, a millionth of a second.
It blinks into existence and reacts almost instantly. which means it doesn't travel very far from where it was created in the mitochondria.
A microscopic spark flying out of the cellular furnace. I can see how that could be dangerous if it lands on something flammable. But that spark doesn't just disappear, does it? It initiates a chain reaction. Because I see the literature focusing heavily on a second type of ROS, which actually isn't a free radical at all, hydrogen peroxide.
Right.
When I hear that, I immediately think of the brown bottle in my medicine cabinet that I pour on a scraped knee. My cells are actually manufacturing that.
All the time. The superoxide anion is frequently converted into hydrogen peroxide, or H2O2. Unlike superoxide, hydrogen peroxide does not have an unpaired electron. It is sound balance, so it is a non-radical.
Okay, so it's calmer.
Much calmer. Because of that, it is much more stable. If superoxide is a chaotic, flying spark, hydrogen peroxide is like a steady, glowing signal flare. It is less reactive, which gives it a longer lifespan. And, crucially, it is membrane permeable.
Meaning it can travel. It's not trapped in the mitochondria where it was made.
Precisely the opposite. It can float away from the mitochondria, slip right through the cellular membranes, and travel to entirely different compartments of the cell. It can even exit the cell and travel to neighboring cells.
That's wild.
It is. This mobility and stability allow hydrogen peroxide to act as a critical messenger, transmitting information across the biological landscape.
A signal flare that can travel through walls. I love that visual. But if we just had the spark and the flare, ROS might not have earned such a terrible reputation. It's what happens when things get out of control that leads to the true villain of this story.
Yeah.
The literature describes something called the hydroxyl radical, and the papers describe it as the most cytotoxic, the most deadly of all ROS. How does a relatively stable signal flare turn into a cellular disaster?
That disastrous transformation happens through a pair of chemical reactions known as the Haber-Weiss and Fenton reactions. And it essentially comes down to a collision with heavy metals.
Heavy metal, like
Your body naturally contains transition metals, specifically iron and copper ions, which are necessary for various biological functions. But they must be tightly controlled. When that stable hydrogen peroxide molecule encounters a free iron ion in the right environment, a violent chemical reaction occurs. The iron strips the hydrogen peroxide apart, generating the hydroxyl radical.
Let me stop you there. So iron, the stuff we need in our blood to carry oxygen, the stuff we take supplements for, is actually the catalyst that turns the signal flare into a bomb.
It can be, if the iron isn't safely bound to proteins. Free iron is incredibly dangerous for this exact reason. To continue our analogy, if superoxide is a spark and hydrogen peroxide is a signal flare, the hydroxyl radical is a highly destructive, uncontrollable, 5-alarm chemical fire.
A 5-alarm fire.
It is wildly reactive. It doesn't travel. It simply destroys the very first biological molecule it touches the microsecond it is formed. There is no enzymatic defense against it. Your body has no specialized fire extinguisher for the hydroxyl radical.
Once it's there, the damage is done.
Exactly. Once it is generated, it causes severe, irreversible damage to your cellular infrastructure.
And that profound damage is exactly what led to the villain era of free radicals. Let's rewind the clock a bit. If we go back to 1956, a scientist named Denham Harman proposed what became one of the most famous biological concepts of the 20th century, the free radical theory of aging. What was he seeing that led him to that conclusion?
You have to consider the scientific context of the 1950s. Radiation biology was a massive field of study following World War II. Scientists were observing how radiation destroyed cells, and they realized that radiation was causing damage by splitting water molecules in the body to create
hydroxyl radicals.
You guessed it, hydroxyl radicals. Harman looked at this and made a brilliant leap. He knew our bodies were naturally producing these free radicals through normal metabolism. He proposed that aging wasn't a programmed biological clock ticking away, but rather the cumulative result of a lifetime of this microscopic rust.
Wear and tear.
As we live, breathe, and eat, our engines produce these sparks and fires. Over decades, that oxidative stress destroys the cell from the inside out.
It makes complete logical sense. You run an engine long enough, the exhaust and the heat eventually degrade the parts. And the literature outlines the specific damage this rust causes when it overwhelms a cell. I'm looking at 3 catastrophic forms of destruction here. Let's start with the outer layer of the cell.
When oxidative stress overwhelms a cell, the first major target is the cell membrane. Your cell membranes are made of lipids, which are essentially structural fats. ROS, particularly those hydroxyl radicals, attack these lipids in a process called lipid peroxidation.
They just burn through the fat.
They literally tear the protective walls of your cells apart. Think of leaving a stick of butter out in the sun until it goes rancid. That is lipid peroxidation happening on a macro scale. In the lab, we can actually measure this cellular destruction by looking for a biomarker called malondialdehyde. It's a chemical footprint left behind when a cell wall is burned by ROS.
So the walls of the house are melting. But the damage goes deeper than that, right into the workers inside the house.
The second major target is the proteins. Proteins are the blue-collar workers of the cell. They do everything from providing structural support to acting as enzymes and chemical messengers. ROS cause protein oxidation, physically altering their 3D shape.
And in biology, shape is everything, right?
Shape dictates function. If a protein is oxidized, it misfolds, it clumps together with other broken proteins, and it completely loses its ability to do its job. We see this clinically as an accumulation of carbonylated proteins. It's like taking a beautifully complex origami swan and crushing it in your fist. It's still made of paper, but it can't function as a swan anymore.
The walls are melting, the workers are striking. And then we have the third target, the genetic blueprint itself.
DNA damage. This is the most terrifying aspect of oxidative distress. Those hydroxyl radicals can smash right into the nucleus of the cell, interacting with the DNA strands. They cause literal physical breaks in the double helix and force mutations in the genetic code.
They're rewriting the manual.
In the worst way possible. When a hydroxyl radical oxidizes a specific part of your DNA, it leaves behind a distinct biomarker known as 8-oxoguanine. This is the hallmark of oxidative genetic damage. It's like someone sneaking into the factory and randomly erasing or changing words in the master instruction manual. This is the exact kind of damage that leads directly to cellular senescence.
Where the cell turns into a zombie?
Yes, or worse, cancer.
Listening to that cell walls melting like rancid butter, worker proteins crushing into useless shapes, the genetic manual being shredded, it's incredibly bleak. It sounds like our cells are just helpless wooden houses waiting to inevitably burn down. But the reality is human beings wouldn't survive a single day, let alone 80 years, if we didn't have an evolutionary countermeasure.
We aren't helpless.
No. The literature details an incredibly sophisticated built-in cellular fire department.
Life evolved on this planet in an oxygen-rich environment. Over billions of years, it developed a multi-layered, brilliantly coordinated antioxidant defense system to fight back against this inevitable exhaust. I want to be clear, this isn't about drinking blueberry juice or eating acai bowls.
Right, this is internal.
This is intrinsic, heavy-duty enzymatic machinery coded directly into your DNA. The first responder on the scene is an enzyme called superoxide dismutase, or SOD.
The first responder. What does SOD actually do when it encounters that chaotic spark we talked about?
Whenever that rogue spark, the superoxide anion, is generated, SOD is stationed right there to catch it. SOD catalyzes a very rapid reaction that dismutates or converts that dangerous superoxide into the much more stable hydrogen peroxide, snuffs the spark, and turns it into the flare.
Okay, so the immediate threat of the spark is neutralized. But now, as we established earlier, you have all this hydrogen peroxide floating around. It's stable, but if it bumps into a free iron ion, you get the catastrophic hydroxyl radical fire. So the fire department's job isn't finished yet.
Far from it. Once SOD converts the stark into a flare, the secondary units of the fire department arrive. These are specialized enzymes, primarily known as catalase and glutathione peroxidase, or GPX. These enzymes have one singular job: safely detoxify the hydrogen peroxide before it can find any iron.
How do you do that?
Catalase, for instance, is one of the most efficient enzymes in the human body. It grabs that hydrogen peroxide and rapidly breaks it apart into two completely harmless substances: plain water and oxygen. Glutathione peroxidase does something very similar, using a molecule called glutathione as a helper to reduce the peroxides into water.
It's a perfectly orchestrated relay race. The engine leaks a dangerous spark, SOD catches it and turns it into a flare. Catalase and GPX catch the flare and turn it into water. As long as this system is balanced, the cell remains perfectly healthy and the house doesn't burn down.
Exactly. But it is that very balance that we misunderstood for so long. We assumed that aging and disease were simply a matter of the sparks eventually overwhelming the fire department.
And that beautifully orchestrated system and our misunderstanding of it brings us to the most fascinating turning point in this whole narrative. We spent decades assuming ROS were just toxic garbage, which led to a very logical but ultimately completely flawed societal conclusion. This brings us to the active debate, where modern science profoundly disagrees with the assumptions of the past 50 years. If ROS are so terrible, the logic went, Shouldn't we just flood our bodies with antioxidants?
That was the thought process, yes.
I vividly remember the 1990s and 2000s. The supplement industry exploded. Vitamin C, vitamin E, beta-carotene. I used to spend $15 a bottle on these massive mega antioxidant smoothies after a marathon, thinking I was saving my cells from rust. The thought was, let's just wipe out all the free radicals and we'll cure aging. But looking at the massive clinical trials over the last 20 years, that didn't happen, did it?
The failure of blanket antioxidant supplementation is one of the most sobering twists in modern medical history. Researchers organized massive, multi-year, double-blind clinical trials. They gave tens of thousands of people high doses of nonspecific antioxidant pills. The prevailing hypothesis was that they would see massive drops in heart disease, cancer, and overall mortality.
And what were the actual results?
The results were largely neutral. People taking the pills didn't live any longer or healthier than the placebo groups, but in some highly publicized cases, the results were actually detrimental. High doses of certain antioxidants were correlated with worse outcomes.
Worse.
Yes. One of the most famous was a trial looking at beta-carotene supplements in heavy smokers. The researchers thought the antioxidant would protect the smoker's lungs from the oxidative stress of the smoke. Instead, they had to halt the trial early because the group taking the beta-carotene was developing lung cancer at a significantly higher rate than the placebo group.
Wait, hold on. You're telling me that giving a massive dose of an antioxidant, the supposed good guy, actually accelerated the cancer? That breaks everything we've talked about so far. Why would that happen?
The scientific community was forced to step back and ask that exact question. And the answer they found completely rewrote redox biology. They realized that wiping out all ROS with massive doses of exogenous antioxidants is like cutting the communication lines to your own cellular fire department. We go back to the idea of the signal flare. The signal flare isn't just a threat waiting to happen, it's an actual message.
So by flooding the body with antioxidants, we were effectively putting out the flares before the fire department even knew there was a problem.
Precisely. This birthed the concept of oxidative eustress versus oxidative distress. We already talked about distress, the severe damage that causes DNA mutations, lipid peroxidation, and cell death. But eustress, meaning good stress, is the profound realization that low to intermediate levels of ROS are not biological accidents. They are vital, indispensable physiological signals.
It's all about balance.
Our cells operate in what researchers call the Goldilocks zone. Too much ROS, and the cell burns down. Too little ROS because you've artificially scavenged them all with pills, and the cell goes completely deaf. It loses the ability to communicate, adapt, and survive.
Here's where it gets really interesting. Because if ROS were truly just metabolic accidents, just random leaks from the mitochondrial engine, why would evolutionary biology design biological machines whose sole dedicated purpose is to produce them on demand? That completely shatters the accident theory. And the research points directly to something called the NOx family of enzymes. Let's talk about them.
The discovery of the NOx, or NADPH oxidase family of enzymes, was a monumental paradigm shift. These are highly specialized enzymes that sit on the cell membranes. They don't produce energy, they don't build cellular structures. Their only job, their entire reason for existing, is to intentionally grab oxygen and turn it into reactive oxygen species. They're intentional ROS generators.
Let me play devil's advocate here. Why on earth would a cell intentionally create the very toxic chemical that is supposed to destroy it? That sounds like a terrible evolutionary design.
It sounds counterintuitive until you realize that the cell uses ROS as a localized chemical weapon and as an essential signaling mechanism. A perfect example is your immune system fighting an infection. When a macrophage, a type of specialized white blood cell, encounters an invading bacteria, it doesn't just swallow it and digest it nicely.
It goes on the offensive.
Exactly. It engulfs the pathogen into a tiny cellular compartment, and then it activates those NOx enzymes. The NOx enzymes unleash what is called a respiratory burst. They intentionally generate massive amounts of superoxide and hydrogen peroxide to chemically burn the invading pathogen to death inside that isolated compartment.
So it's literal chemical warfare happening inside you, on your behalf. My immune system is using the rust to burn the invader.
Without ROS, your immune system would be entirely defenseless against bacterial infections. You would succumb to the first minor cut you received. But it's not just about killing invaders. These NOx enzymes are also essential for creation and healing. The literature shows that ROS regulates cellular proliferation, the differentiation of stem cells, and even the regeneration of damaged tissue. You literally cannot grow or heal without these reactive molecules sending the signal to do so.
Give me a real-world example of that. How does a toxic spark
Help me heal a wound. Let's say you cut your finger. Immediately, the cells at the edge of that wound intentionally generate a burst of hydrogen peroxide. That hydrogen peroxide diffuses outward and acts as a beacon. It alerts the immune system, recruiting white blood cells to clean up the microscopic debris, but it also signals the surrounding healthy skin cells, telling them, we have a breach, you need to start dividing and rebuilding the tissue.
And if you interfere with that?
If you were to somehow flood that specific wound with incredibly potent, indiscriminate antioxidants, you would actually impair the healing process because you are hiding the biological signal. The cells wouldn't know they needed to divide.
Which perfectly explains the fascinating and frankly hilarious relationship between ROS and a physical exercise. We have this biological principle called hormesis, the idea that what doesn't kill you makes you stronger. A low dose of a biological stressor triggers a beneficial adaptation. Exercise is the ultimate example of this. When I go for a run or lift heavy weights, my metabolic rate skyrockets. My mitochondria are working in overdrive, breathing heavier, pumping harder, and as a result, they produce drastically more ROS. I am creating a massive temporary spike in oxidative stress.
And under the old, flawed paradigm of the 1980s, that spike in ROS during a workout should be terrible for you. It should be causing rapid aging and tissue destruction. But we know empirically that exercise is arguably the single best thing you could do for your health span and longevity. Why? Because that temporary, intense spike in ROS is the exact, precise signal your body needs to adapt.
It's the trigger.
Yes, the ROS flood out of the mitochondria and signal your muscle cells to build more mitochondria to handle the load next time. They signal the cells to increase insulin sensitivity and to grow stronger, denser muscle fibers. But most importantly, that temporary spike in ROS signals your DNA to drastically ramp up the production of your own intrinsic antioxidant defenses. You are telling your body to build more SOD, more catalase. You are training your internal fire department to be stronger, faster, and more efficient.
And this is where the supplement craze backfires spectacularly, and where I realized I was wasting my money in my 20s. There have been incredibly revealing studies where researchers put people on rigorous exercise programs, but gave half of them massive doses of antioxidant pills, like 1,000 milligrams of vitamin C and large doses of vitamin E, right after their workouts. What actually happened to the people taking the pills?
The people taking the massive antioxidant pills completely blunted the beneficial adaptations of the exercise. Their muscles didn't adapt as well, their physical endurance didn't improve as much, and their baseline insulin sensitivity didn't get the same boost as a placebo group.
They completely sabotage themselves.
By artificially suppressing the ROS spike with high-dose vitamins, they erased the chemical text message that tells the body it just worked out. They stole the physiological benefits of their own hard work. This perfectly illustrates the profound danger of viewing ROS simply as a toxin. When you arbitrarily wipe them out, you're interfering with the fundamental language of human physiology.
That is just mind-blowing. The very thing we tried to eradicate is the messenger telling our bodies to get stronger. It really makes you second-guess reaching for that mega-dose vitamin water after a gym session. So if we accept that ROS are vital communicators, we have to look at how exactly they transmit these messages. The cell is huge compared to a single molecule of hydrogen peroxide. Who is receiving the text message? Which brings us to our third major area of focus, frontiers, and what might change everything we know about medicine. We are diving deep into the master regulatory pathways of the cell. If ROS is the messenger, who is the receiver?
What's fascinating here is the sheer mechanical elegance of the biological machinery involved. The primary recipient of these redox messages, the absolute master regulator of our cellular defense and health span, is a pathway known as the KEAP1 NRF2 periFTOR pathway. It is, without a doubt, one of the most important regulatory mechanisms in all of human biology.
We need a good way to visualize this.
To understand it without getting lost in the biochemistry, we need to picture a very specific, almost cinematic relationship between two proteins. Let's start with NRF2.
I like to picture NRF2 as the cellular fix-it guy. He is a transcription factor, which means his job is to go into the nucleus of the cell, open up the DNA master blueprints, and turn on the genes that build our defenses. He's the guy who orders the construction of all those fire department enzymes we talked about earlier, the SOD, the catalase. He has the blueprints to save the cell, but he isn't allowed to just run around turning on genes whenever he feels like it. He has a boss, or rather, a very strict warden.
That's an excellent analogy. The warden is a protein called KEAP1. KEAP1 is a sensor and an adapter protein. Under normal calm conditions, when you are resting on the couch, your heart rate is low, and there is no oxidative stress, the cell doesn't want to waste precious energy building fire extinguishers it doesn't need. So KEAP1 acts as the strict warden. It physically binds to the NRF2 fix-it guy in the main body of the cell, the cytoplasm, completely preventing it from entering the nucleus.
Just keeping him locked down.
But KEAP1 doesn't just hold NRF2 in a cell, it actively condemns it. KEAP1 acts as a structural bridge, linking NRF2 to a complex piece of cellular machinery called a colon-3-based E3 ubiquitin ligase.
Wait, a colon-3 base, what is a ligase doing here? Break that down for me, because that sounds like heavy machinery.
It is heavy machinery. The colon-3 ligase is essentially a tagging system for the cellular garbage disposal. Its job is to slap a specific chemical tag, called ubiquitin, onto the NRF2 protein. Once NRF2 has this ubiquitin tag stuck to its back, it's immediately recognized by the proteasome. You can think of the proteasome as a molecular wood chipper.
A wood chipper, wow.
It grabs the tagged NRF2 and shreds it down into basic amino acids so the parts can be recycled. So under normal, unstressed conditions, the fix-it guy is constantly being produced by the cell, immediately captured by the warden, tagged by the ligase, and thrown into the wood chipper. It's a continuous, rapid modal turnover that keeps the cellular defense system completely turned off.
A brutal, short existence for NRF2. But then an emergency happens. You go for a run, or you encounter a toxin, and oxidative stress occurs. The engine revs up, the ROF levels start to rise in the cell, the signal flares are flying. How does the warden know to stop the wood chipper and let the fix-it guy go do his job?
This is where the brilliant, sensitive chemistry of KEAP1 comes into play. KEAP1 is not just a blind warden holding a lock. It is an incredibly sensitive chemical sensor. Embedded on the surface of the KEAP1 protein are highly reactive, highly sensitive cysteine residues.
Cysteines.
Cysteines are amino acids that contain sulfur, and they act like delicate pressure plates. They're incredibly sensitive to oxidation. When ROS levels spike, those hydrogen peroxide molecules floating through the cell physically interact with and oxidize these specific cysteine residues on the KEAP1 protein.
So the signal flare literally hits the warden directly.
It hits the warden, and the impact is profound. When those cysteine residues are oxidized, it forces the entire KEAP1 protein to physically change its shape. It undergoes a massive conformational change. Because its physical shape is warped, KEAP1 can no longer properly hold on to NRF2, and it can no longer align it with the colon-3 ubiquitin ligase. The tagging process completely stops. NRF2 is no longer sent to the wood chipper.
The warden drops his keys. The fix-it guy is liberated.
Liberated, and it rapidly floods into the nucleus of the cell. Once inside the safety of the nucleus, NRF2 searches the DNA for very specific sequences called antioxidant response elements, or AR. When it finds an ARE, NRF2 binds to it and essentially flips a massive master switch. It activates a huge battery of cytoprotective genes.
What kind of genes are we talking about?
We are talking about phase 2 detoxification enzymes, proteins like hemioxidase-1 and QO1, and massive upregulations of glutathione production. The cell goes into full biological lockdown, building a fortress of antioxidant defenses to neutralize the toxic threat, restore the balance, and repair whatever damage the initial sparks caused. And here's the beautiful part. Once the ROS levels drop back to normal because the fire department put the fire out, the cysteines on KEAP1 are reduced back to their normal state. KEAP1 regains its original shape, grabs the keys, and starts grabbing NRF2 and sending it to the proteasome again. The crisis is averted, and the system powers down to save energy.
It's an astoundingly elegant, self-regulating feedback loop. And what the latest research is showing us is that this isn't just happening randomly all over the cell in a big, messy biochemical soup. It's highly organized. The science of subcellular compartmentalization proves that it's not just about how much ROS is produced, but where it is produced. Location, location, location. So if ROS are sending these signals, how does the whole cell not just catch on fire? How is the signal contained so it doesn't cause a false alarm everywhere else?
The spatial dynamics of ROS are the absolute new frontier in redox biology. For a long time, we pictured the cell as a balloon filled with water where chemicals just float around and bump into each other randomly. That couldn't be further from the truth. The cell is a highly structured, densely packed city with specific neighborhoods and highways. A ROS signal generated at the cell membrane doesn't flood the whole cell. It is contained within structures we now call redoxosomes.
Redoxosomes? That sounds like a specialized containment unit.
It is exactly that. A redoxosome is a tiny membrane-bound vesicle. When a cell receives a signal at its outer membrane, it pinches off a tiny bubble of that membrane and pulls it inside. This bubble, the redoxosome, contains a specific receptor and an NOx enzyme. The NOx enzyme creates a highly localized, contained burst of hydrogen peroxide that oxidizes a very specific target protein right next to it. completely altering that protein's function without leaking out and affecting the rest of the cell.
So it really is a direct text message to a specific phone, not a global emergency broadcast over a loudspeaker.
Exactly. Another prime example is mitochondrial ROS. We used to think these sparks just leaked out and caused indiscriminate damage. Now we know that mitochondria form actual physical contact points with the endoplasmic reticulum, which is another massive organelle that stores calcium.
Mm.
These contact points are called mitochondria-associated ER membranes, or MAMs. The mitochondria intentionally release precise pulses of ROS at these MAMs.
And what did those pulses do?
The ROS acts as a switch, telling the endoplasmic reticulum to release calcium. That calcium flows back into the mitochondria, which in turn tightly controls the cell's ATP energy production. ROS is functioning as a highly targeted molecular switch, turning energy production up or down based on immediate demand. It's a level of biological engineering that is truly awe-inspiring.
It is awe-inspiring, but it also raises a terrifying question. So what does this all mean when things go wrong? Because if this system, the warden, the fix-it guy, the precise text messages, is so perfectly designed to protect us, why do we still get sick? Why is cancer still such a pervasive, unstoppable threat in society? And that leads us to the dark side of this exact pathway. We just talked about NRF2 as the ultimate biological hero, the fix-it guy who builds our defenses and prevents damage. But what happens when a tumor figures out how to hijack the hero?
It is one of the most insidious, brilliant tricks in all of cancer biology. In normal, healthy tissue, NRF2 is your absolute best defense against cancer, because it prevents the oxidative DNA damage that causes the initial mutations in the first place. But once a cell has already become cancerous, the rules of the game completely change. Tumor cells exist in a very harsh, hostile microenvironment. They are rapidly dividing out of control, they are generating massive amounts of toxic metabolic waste, they are constantly being attacked by your immune system, and because they outgrow their blood supply, they are often starved of oxygen. To survive this brutal environment of their own making, cancer cells develop what we call a redox addiction.
They realize they are constantly on fire, so they need a permanent shield. They need infinite fire extinguishers.
Exactly. And they get that shield by mutating the very pathway we just described. In many aggressive cancers, such as non-small cell lung cancer, the tumor cells undergo somatic mutations in the genes that code for either the KEAP1 warden or the NRF2 fix-it guy. A mutation in the KEAP1 gene might permanently destroy the warden's ability to bind to NRF2, or a mutation in the NRF2 gene might change its shape just enough so the warden can't recognize it. The result is the exact same. The warden is permanently disabled. NRF2 is never sent to the wood chipper. It floods the nucleus and stays there permanently, 24/7. We call this the constitutive activation of NRF2.
The master switch is literally taped down in the on or end position.
Yes. The tumor cell starts pumping out massive, continuous levels of antioxidant enzymes. It builds an impenetrable biological armor. And why is this so devastating for the patient? Because almost all of our most effective cancer therapies, radiation therapy, and traditional chemotherapies work by intentionally generating massive amounts of ROS to overwhelm the tumor cell and force it to undergo apoptosis, or programmed cell death. But if the tumor has hijacked NRF2, it has an infinite supply of fire extinguishers.
So the treatment just bounces off.
You hit the tumor with radiation, generating a massive ROS fire, and the tumor's hijacked NRF2 system instantly puts the fire out. The cancer becomes incredibly resistant to chemotherapy and radiation. The very evolutionary system designed to protect us is twisted and weaponized to protect the tumor.
That is a chilling realization. The tumor weaponizing our own defense mechanisms against our treatments. It perfectly highlights how delicate this balance is. Which naturally brings us to our final section, implications. We need to connect this microscopic, subcellular science back to the macroscopic world you and I live in. How does this dictate the future of disease, aging, and the economy of medicine? Because the stakes couldn't be higher. Let's step away from cancer for a moment and look at the #1 killer globally, cardiovascular disease. How does this ROS story play out in our arteries to cause a heart attack?
If we connect this to the bigger picture, the pathology of atherosclerosis, which is the hardening and clogging of the arteries, is essentially a masterclass in chronic oxidative distress. It doesn't start with fat. It starts with ROS. It all begins at the vascular endothelium, which is the delicate, single-cell thick inner lining of your blood vessels. Under healthy conditions, your endothelial cells constantly produce a gas called nitric oxide, or NO. Nitric oxide is a crucial vasodilator. It keeps the blood vessels relaxed, wide open, and slippery, ensuring blood flows smoothly and preventing clots from forming.
But then modern life happens. The risk factors pile up. High blood pressure, a diet high in processed foods, smoking cigarettes, chronic stress, all of these things trigger a localized storm of oxidative stress right there in the blood vessel wall.
Exactly. Massive amounts of superoxide are generated by NOx enzymes and something called uncoupled endothelial nitric oxide synthase in the blood vessel wall.
Whoa, hold on, uncoupled. Break that down for me. What does it mean for an enzyme to become uncoupled in my blood vessel?
It's a great mechanical analogy. The enzyme that normally makes the helpful nitric oxide, endothelial nitric oxide synthase, requires a specific helper molecule to function correctly. When you are under severe oxidative stress, that helper molecule gets destroyed. Without its helper, the enzyme uncouples. It acts like an engine that is thrown a timing belt. Instead of producing the helpful nitric oxide, the broken engine starts pumping out massive amounts of toxic superoxide sparks instead.
Oh, wow. So the machine designed to keep the blood vessel relaxed literally breaks and starts shooting sparks into the wall.
Yes, and it gets worse. That superoxide has a vicious, immediate chemical reaction with whatever nitric oxide is left. It literally binds to it and destroys it, forming a highly toxic molecule called peroxynitrite. Instantly, you have severely reduced the bioavailability of nitric oxide in that area. The blood vessel can no longer relax, it constricts, driving your blood pressure even higher, but that's just the mechanical beginning. The most critical, deadly event happens to your cholesterol.
We've all heard of LDL cholesterol, the bad cholesterol. My doctor tests for it every year. But standard LDL floating in your blood isn't actually what causes the plaque, is it? It's when that LDL interacts with the sparks. It's when it gets oxidized.
Correct. Standard LDL is just a delivery vehicle for fat. But when it floats through that inflamed, spark-filled blood vessel wall, the ROS penetrate the LDL particles. They oxidize the lipids and proteins inside them, creating ox-LDL.
Yeah.
Your immune system does not recognize ox-LDL as normal, healthy cholesterol. It recognizes it as a dangerous, toxic, foreign invader. So it sounds the alarm. It sends in those exact same macrophages we discussed earlier, the ones that eat bacteria, to clean up the mess in your artery. The macrophages flood into the blood vessel wall and start eating the ox-LDL. But there is a fatal flaw in the macrophage's biological programming. It doesn't have an off switch for eating fat. It consumes so much oxidized fat that it literally becomes bloated, swollen, and immobilized, turning into what pathologists call a foam cell.
Because under a microscope, a collection of them looks like sea foam,
Mm-hmm.
and they are just packed full of toxic, rancid fat.
And eventually, these foam cells eat themselves to death. They rupture, spilling this necrotic, toxic, highly oxidative core of fat and cellular debris right into the wall of your artery. This triggers a massive chronic inflammatory cascade. Your body panics. Smooth muscle cells migrate to the area to try and wall off the toxic dump, creating a thick fibromuscular cap over it, like putting a scab over a festering wound. This is the atherosclerotic plaque. Over years and decades, as oxidative stress continues to weaken the structural integrity of that cap, it can suddenly rupture. When it ruptures, it exposes that highly thrombogenic core to your blood, triggering an instant massive blood clot. If that clot blocks blood flow to the heart muscle, it is the myocardial infarction, a heart attack. If it breaks off and blocks blood to the brain, it is an ischemic stroke. It all traces back to that initial loss of redox balance, those initial sparks in the endothelium.
Which means if we can figure out how to intervene at the redox level, before the LDL ever oxidizes, or before the macrophages turn into foam cells, we could potentially stop the #1 killer of human beings in its tracks. And that is the promise of the future. The literature points toward a massive shift called precision redox medicine. As we've clearly established, we can't just throw a bucket of vitamin C at the problem. We have to shift from this outdated, clumsy idea of globally scavenging ROS to a new paradigm of therapeutic redox reprogramming. We have to be biological snipers, not carpet bombers. And the clinical trials happening right now to achieve this are breathtaking. Let's talk about the efforts to deliberately pharmacologically control that NRF2 master switch. How do we send a drug to tell the warden to release the fix-it guy?
This is where pharmacology is making incredible, highly targeted strides. Researchers have developed highly specific molecules designed to be NRF2 activators. They belong to a new class of drugs known as antioxidant inflammation modulators, or AIMs. The most prominent example currently in advanced clinical trials is a drug called bardoxolone methyl. The chemistry behind it is brilliant. Bardoxolone methyl is an electrophile. Its entire chemical structure is designed to intentionally target and bind to those specific, highly sensitive cysteine residues on the KEAP1 protein we talked about earlier.
So the drug is literally mimicking the signal flare. It binds to the warden, forces him to change his physical shape, and makes him release NRF2 without ever needing actual oxidative stress to trigger it.
Precisely. It tricks the warden. It's currently being tested in phase 3 clinical trials for patients with advanced chronic kidney disease, particularly those with type 2 diabetes. These patients suffer from severe, relentless oxidative stress and inflammation in their kidneys, which slowly and painfully destroys the organ's intricate filtering system. By administering bardoxolone and methyl, doctors are artificially flipping the NRF2 master switch in the patient's kidney cells. They are commanding the cells to drastically upregulate their own intrinsic antioxidant and anti-inflammatory defenses. It is fundamentally different from giving a patient an antioxidant pill. You aren't giving them the fire extinguisher. You're reprogramming the patient's own DNA to build up a better fire department, to flint the disease from the inside out.
That is just incredible. We are hacking the cellular software. But pharmacology is only one avenue. If we look slightly further over the horizon, the literature touches on emerging technologies that sound like pure science fiction. Let's look at gene therapy. With the advent of CRISPR-Cas9, we aren't just limited to drugs that interact with proteins. We can actually edit the blueprint of the factory itself. How does CRISPR fit into redox biology?
With CRISPR, the possibilities for redox medicine are staggering, though they require immense precision. Instead of relying on a daily drug to activate a pathway, we could theoretically use gene editing to permanently upregulate or fine-tune RAS-related genes. For example, editing a specific cell lineage to produce higher baseline levels of NQO1, a powerful detoxification enzyme, or modifying the p53 tumor suppressor gene to better manage oxidative stress responses in individuals who are genetically predisposed to certain types of cancers. We could essentially tweak the thermostat of the cellular fire department to run exactly at the optimal temperature for an individual's unique biology and risk factors.
And then there's stem cell therapy. We're seeing trials using mesenchymal stem cells, or MSCs, to treat tissues that have been severely damaged by oxidative stress, like a heart after a massive infarction or a lung scarred by chronic disease. What makes an MSC so special in a redox context? I always thought stem cells just turned into new tissue to patch a hole.
That's a common misconception. MSCs are fascinating because they don't primarily function by just physically replacing damaged cells. They act more like mobile biological pharmacies. They are factories that secrete a potent, localized cocktail of healing factors. When you introduce MSCs into an environment of high oxidative distress, like a heart muscle immediately following a heart attack, they sense the extreme damage. In response, they secrete specific antioxidant cytokines and growth factors that actively neutralize the toxic environment, calm the screaming immune system, and rescue the dying tissues around them. They actively reset the local redox homeostasis.
It's like deploying a specialized hazmat team to chemical spill. They don't just rebuild the building, they clean up the toxic sludge so the local workers can return. And perhaps the most counterintuitive yet brilliant application of this new understanding is how we are reinventing cancer treatments. We just spent a long time talking about how tumors hijack NRF2 to become resistant to chemotherapy. So researchers are exploring something called antioxidant chemotherapy. I have to admit, when I first read that, it sounded like an absolute oxymoron based on everything we've discussed, but it's actually a stroke of genius in precision targeting. Walk me through how this works.
It really is an elegant solution to a brutal problem. Let's take a traditional heavy-hitting chemotherapy drug like cisplatin. Cisplatin is highly effective at killing tumors. It works by entering the cell, binding to the DNA, and inducing massive, overwhelming oxidative stress and DNA damage until the cell commits suicide. The massive problem in oncology is that cisplatin causes the exact same oxidative damage to healthy tissues. This leads to severe, sometimes permanent side effects. It causes profound nephrotoxicity in the kidneys. It causes ototoxicity, meaning the ROS literally destroy the delicate hair cells in the inner ear, leaving patients permanently deaf. The new approach is to combine the chemo drug, cisplatin, with a targeted systemic antioxidant like N-acetylcysteine or NEC.
But wait, I'm playing proxy for the audience here. Wouldn't the NEC just put out the fire in the tumor too? Wouldn't it protect the tumor from the cisplatin, rendering the chemo useless?
That's exactly what everyone feared, and that's the genius of the specific protocol. It all comes down to the dosing, the timing, and the incredibly different metabolic rates of the tissues involved. A tumor is metabolizing and dividing at a furious, uncontrollable rate. It takes up massive amounts of whatever's in the blood, including the cisplatin, very quickly. The normal, healthy tissues, like the inner ear and the kidneys, metabolize much slower. In these highly calibrated combination therapies, the goal is to provide just enough NAC to protect the flow-metabolizing normal tissues from the oxidative collateral damage. The NAC provides a temporary shield for the ear and kidney. Meanwhile, The rapidly dividing tumor takes up such a massive dose of the cisplatin so quickly that the ROS storm inside the tumor still completely overwhelms any protection the NAC might have offered.
So you are essentially giving the healthy civilian cells a bulletproof vest while still dropping the bunker buster bomb on the cancer.
Precisely. It's all about threading the needle. You are manipulating the redox thresholds of different tissues simultaneously.
Which demands an incredible, unprecedented level of precision from the physician. You can't just guess where a patient's threshold is.
This raises an important question, perhaps the most important question in modern redox medicine. How do we actually know what an individual patient's redox state is? If the line between eustress and distress is so incredibly thin and varies wildly from person to person, we can't just guess. We can't manage what we can't measure. We can't just look at a global marker of oxidative stress in a blood draw and assume that tells us what is happening in the liver or the brain. We need highly specific real-time spatial biomarkers. We need to map the exact redox state of specific organs. And that is where tools like the incredibly groundbreaking oximouse model come in.
The oximouse. I read this paper and my jaw dropped. This is a tremendous breakthrough from researchers at Harvard. They have essentially managed to map the exact oxidation state of every single tissue in a living, breathing mouse. Explain how this changes the game for us.
The oximouse is a comprehensive quantitative map of the cysteine oxidation networks across different tissues in vivo. What the researchers did was figure out a way to chemically tag and measure exactly how oxidized those sensitive cysteine proteins are in real time across the entire body. What this model revealed completely shattered old assumptions. It proved that redox networks are highly tissue-specific. The baseline oxidative state, the normal, healthy weather of a liver cell, is completely different from the weather in a brain cell, which is different from a heart muscle cell.
So my liver might run hot and my brain might run cool, naturally.
Yes. Furthermore, these networks change uniquely as the organism ages. The liver might become more oxidized over time, while another organ might remain stable. This means that a one-size-fits-all redox therapy is biologically impossible and potentially dangerous. Your liver might be in a state of severe oxidative distress, desperately needing intervention, while your skeletal muscles are in a perfect state of oxidative eustress, happily adapting to exercise. The future of precision redox medicine relies on this exact concept. The goal is to take a patient, run a comprehensive metabolic profile to understand their exact tissue-specific redox signature, and then tailor a highly specific intervention. Whether it's administering an aimed drug like bardoxolone, executing a targeted CRISPR edit, or utilizing an antioxidant chemo combo, the goal is to push their specific physiology back into their unique Goldilocks zone.
It is a totally new frontier. We are moving from blunt, primitive instruments like throwing handfuls of vitamin C at the wall to see what sticks, to using microscopic molecular scalpels. We are learning the actual language of the cells. And when you step back from the massive stack of papers we've gone through today, when you look at the entirety of this story, from the electron transport chain to the oximouse, you are struck by the ultimate, beautiful irony of life itself. You and I spend our entire lives breathing in oxygen. It is the invisible fuel that allows us to laugh, to run marathons, to think complex thoughts, and to love. We literally cannot survive for more than a few minutes without it. Yet the exact same molecule is the very thing that slowly, relentlessly rusts our cells from the inside out over a lifetime. It is the absolute giver of life and the inescapable arbiter of our aging. But what we've learned today is that aging isn't just about accumulating that rust. It is so much more elegant and, frankly, so much more tragic than a simple mechanical breakdown. Aging is a failure of our internal cellular communication network. It is the slow, gradual silencing of those precise, whispering chemical signals. The flares lose their brightness, the warden gets tired, the fix-it guy forgets his blueprints, the network loses its fidelity over time. The key to a longer, healthier life isn't about turning the signals off, burying them in antioxidants, or fearing the oxygen we breathe. It's about learning how to listen to them, how to tune the cellular radio back to the right frequency. And it leaves you wondering something truly profound. If we eventually map this entire network, if we learn to perfectly control these microscopic signal flares with CRISPR and AIMS, could we do more than just pause aging? If the cells are simply waiting for the right text message to heal, to divide, to rebuild, maybe immortality isn't a mythical fountain of youth. Maybe it's just a perfectly coded chemical text waiting for us to hit send. Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support. And to our listeners, thank you for being part of this journey with us. We hope you enjoyed today's discussion. If you did, be sure to subscribe and support us on Patreon. And of course, you can write to us in comments if a specific topic needed to be covered, which help us all to grow. Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.